Izzy and Nola were having a nice conversation about Anatomy 2 and how lovely of a teacher Mr. Roach is, when Izzy sneezed and didn't cover her mouth. Unfortunately for Nola, the virus is looking for a new host to invade, but in order to do so, the virus will have to get past the first defense system of Nola's lymphatic system, which is the nonspecific defense system. The virus must begin by breaking through the first physical nonspecific barrier, which is the skin, and enter Nola's body through the opening in her nose. Once the virus enters Nola's nose, it has to get past the hair and secretions within her nasal cavity. The virus also must escape the sticky mucous membranes lining all of the exposed surfaces of Nola's cavities. If the virus manages to get past these physical barriers, then it is considered an infection. After the virus successfully bypasses Nola's physical barriers, it hijacks the body cell's replication cycle, hiding and posing as a self-cell, masking itself from phagocytes. Phagocytes include macrophages and neutrophils and are the body's first line of defense once the physical barriers have been broken. Macrophages can be fixed or free large cells that are derived from monocytes. They engulf and digest foreign particles and cellular debris, helping to clean up the body. Neutrophils, in addition to being a crucial phagocytic cell, are classified as microphages, which also include eosinophils and basophils. Neutrophils are the most abundant and aggressive phagocytic cell, quickly ingesting cellular debris or invading bacteria until they explode, forming pus. Eosinophils target foreign compounds or pathogens that have already been marked by antibodies, and basophils release heparin to prevent clotting as well as histamine to increase inflammation. They also work with mast cells to produce the same inflammatory response. Inflammation response is another part of the body's nonspecific defense system. It produces local swelling, heat, redness, and pain in response to any stimulus that damages cells. Inflammation is used to stimulate cell replication, slow the spread of pathogens from injury sites, and mobilize the immune response, aiding in tissue repair. Another part of Nola's nonspecific defense is immunological surveillance, which involves natural killer cells who scan the body for abnormal self cells. NK cells monitor normal tissue in the body and recognize foreign antigens on the membranes of cells or lack of normal receptors. They cause infected cells to go through apoptosis by producing perforins and granzymes to destroy the membranes. They also release cytokines to stimulate macrophages and more NK cells. This last nonspecific defense is a fever, regulated by proteins called pyrogens that cause the hypothalamus to raise the body temperature to above 99 degrees Fahrenheit in order to make the conditions for the virus to live extremely inhabitable. A slight fever is good because high temperatures increase metabolism, allows macrophages to move faster, and stimulates enzymatic reactions. If Nola's nonspecific defense system does not successfully defeat the virus, it then faces Nola's specific defense system. The specific defense system is more targeted, slower to react, and long-lasting compared to the nonspecific defense system. It can be separated into two different responses to antigens: cell-mediated response and humoral response. The two cells that dominate Nola's specific immune response are the B and T cells, both originating as lymphocytes in bone marrow but maturing in different areas of the body. B cells remain and mature in red bone marrow, while T cells migrate and develop in the thymus. Nola's cell-mediated immunity will attack viruses within infected cells and bacteria directly. Antigen-presenting cells such as macrophages play an important role in cell-mediated immunity. Once a macrophage engulfs an antigen, it actually presents a fragment of this antigen within MHC2 on its surface. During this process, interleukin-1 is produced, calling in helper T cells and making Nola feel terrible in order to preserve her energy for the battle ahead. Helper T cells have receptors that bind to peptides presented by the MHC2 on antigen-presenting cells. One of Nola's specific helper T cells is activated by binding to the MHC antigen complex on the surface of the APC with the help of CD4 proteins. Nola's helper T cell then secretes interleukin-2, which partially activates cytotoxic T cells, excites B cells, and stimulates the helper T cell to start dividing. Cytotoxic T cells also play a very important role in the cell-mediated response and help to kill infected cells. Unlike MHC2, which is only found on antigen-presenting cells, MHC1 is found on all nucleated cells and present fragments of proteins synthesized by that cell. However, if the cell is infected, it will present abnormal protein or have an insufficient amount, indicating to the cytotoxic T cell that it needs to be killed. Cytotoxic T cells will kill its target cells mainly by releasing perfin, which forms pores in the target's membrane, causing it to lysis. This process is aided by CD8 protein in order to keep the cells together. Once the cytotoxic T cell is successfully activated, it will kill other cells infected with the same pathogen. NOLA's cell-mediated and humoral immune responses are linked together by cell signaling interactions, especially regarding her helper T cells. Unlike NOLA's cell-mediated response, which included mostly her T cells, her humoral response involves B-cell activation and results from the production of antibodies that circulate in body fluids. This response is initiated when B cells containing antigen receptors bind with specific antigens. However, in order for B cells to fully activate, they must bind with a specific helper T cell as well as receive interleukin-2. Once activated, NOLA's B cells proliferate and differentiate into clones of antibody-secreting plasma cells or effector B cells and clones of memory B cells. The antibodies that effector B cells produce are also known as immunoglobulins. Antibodies function by interacting with a portion of the antigen known as an epitope or antigenic determinant. Antibodies are typically Y-shaped, and the amino acid sequences that make up the V portions of these antibodies vary depending on each specific antibody. There are 5 major classes of antibodies, including IgM, IgG, IgE, IgA, and IgD. IgA attack pathogens before they enter the body. IgD are part of B-cell receptors. IgE helps protect against parasitic worms and is responsible for allergic reactions. IgM are the first on site and responsible for the early stages of immunity. And lastly, IgG are the most diverse, defending against viruses and bacteria and able to cross the placenta. Because NOLA has been infected by a virus, her body will most likely utilize antibody IgG. Antibodies can get rid of antigens through a variety of methods. Neutralization blocks the activity of the antigen. Opsonization enhances macrophage attachment. Agglutination clumps together antigens, preventing them from easily moving around. Precipitation forms immobile precipitates that are disposed of by phagocytosis. With the help of this complex system, NOLA is able to recover from her virus within a few days. But next time she is infected by the same virus, memory B cells will recognize the antigen much quicker, and NOLA most likely won't even become sick to begin with. All better!